Hello and welcome to another wonderful time with DNA. Today we'll be discussing something called the nephron and the renal systems. And some of you are probably wondering, okay, why on earth do you have a picture of an animal in anatomy and physiology picture, and why is it so adorable? Well, first off, this little mouse isn't just any ordinary mouse. It's a Morro Bay kangaroo rat. And what's so special about him and what's so unique about him in our discussion coming up is that he only has to eat seeds, grasshoppers, what have you, in order to survive or get the water that he needs. That's right, he does not have to drink excess water in order to be able to have appropriate life. Whereas us, if we run out of water in like 24 hours, we're down. Now, what we're going to do is we're going to explore how this is possible. First, we're going to go ahead and look into the nephron, that functioning unit, see its varying components, and then we're going to apply what we learned about humans into this little Morro Bay kangaroo rat to make certain we fully understand the nephron. So without further ado, let's go ahead and begin our discussion. So the part of the kidney that is the filtering unit, that is the thing that where the blood interacts and lets go of any stuff, is called the nephron. Now, what's going on here is a lot of things. It has tons of vital roles, just tons upon tons. Okay, not really, more like 5 or 6, but we're going to go ahead and run with it because I like being dramatic. So, first off is the mineral balance. You need a certain balanced amount of sodium, potassium, chlorine, all in your blood, and these nephrons, these kidneys, help out with that. It also does a lot of work of excreting soluble wastes, such as urea and all those things you just don't want around. It also does a lot with blood osmolarity, aka how much does your blood like water, followed by maintaining that blood pH. Finally, if you're going back to AMP3 goodness, the nephron is the part that I didn't mention back in AMP3 because that's a little too much detail. But this is the part that monitors that O2 level if you have enough oxygen. If you don't, it's the part that releases that erythropoietin and sends it out to that bone marrow and says, hey, make more hemoglobin, get it going. Make those cells, make more red blood cells. So this right here is it in all of its cartoonish glory. I do not have a picture of it, but this will definitely suffice. I want to point out that this is the beginning right here. What you're going to see is a glomerulus, aka a bed of capillaries, blood capillaries hanging out inside a Bowman's capsule. Here is where liquid and all that stuff that can sneak past those capillary walls sneak into this nephron, where it can be selected, processed, and excreted if need be. The flow of it is blood into here, filter goes out, goes down these wonderful little tubes, comes out into the collecting duct, all that stuff comes together, goes out your kidney, down your ureter, into your bladder, out your urethra. We'll talk about that in a little while, but let's stay focused on that nephron. So again, hydrophilic filtrate. Remember back that the digestive tract, your liver has a lot more of the excretory function of your hydrophobic part. Your kidneys does the hydrophilic part. So things that increase that filtration rate. Number one is increased water capillary pressure, aka do you have low blood pressure or high blood pressure? Are these arterioles constricted at certain points? Do they force more water out? What have you. If there's a higher blood pressure, you're going to have a lot more filtrate. Also, the amount of surface area those capillaries have. If you have the more surface area, the more places water can leak out. Things that would decrease the filtration rate are as follows. First, going back and eliciting the principles of physics. For every reaction, there is an equal and opposite reaction. If you're pushing stuff out, that stuff that you're pushing out can more than likely come back and push back. So that pressure from that other stuff is pushing back. A couple of others, you got a capillaries less permeable, you seclude it, you decrease that, or you don't permit certain things out, yeah, that's going to affect it. And finally, there's something called a colloidal osmotic pressure. That's just the proteins and other hydrophilic substances' capability of holding on to water. To demonstrate a little bit better, I have a couple of examples. First off, a nice little photo of this poor little pupper is trying to get his stick through the door. Poor little poopers. That stick's just too darn big. So if we were to relate it to our little nephron, the stick would be a plasma protein. The dog would be the water molecule. The outside's the capillary, and inside's the Bowen's capsule. That doggo just loves his stick so much. That water molecule loves its protein so much, but because that protein is just too big, the protein won't go through, but the water molecule just refuses to let go of that stick and it sticks around. So it's stuck in that capillary bed. Another example that was used that's just fantastic at demonstrating the osmotic pressure occurred on the International Space Station. This is a Canadian astronaut that they actually do a little web series in which they field questions. This gentleman was asked what would happen if you put water on a rag and then wrung it out. So he's going to go ahead and demonstrate that now. And he looks very excited about that. Notice that water. That is so cool, ain't it? So notice that water is holding on to his arms, it's holding on to that rag, all that hydrophilic stuff. That's what's going on inside your blood capillaries. That protein, that hydrophilic thing is holding on to that water. Now, let's talk about the different things that affect that filtration rate, those hormones that have a role in it. So for instance, at the glomerulus, again, the glomerulus, the blood capillaries, senses of low blood pressure. What will happen? These cells around these arterioles will release renin, a hormone. That renin will go out, activate angiotensin 1. Angiotensin 1 will activate angiotensin 2. And angiotensin 2 is going to come back, hit that glomerulus, and constrict those arterioles. If it constricts it, that means less blood flow into that, which means even less filtrate's going to come back in or go out into the urine. Thus, if you increase or maintain a volume, you will not have a blood pressure drop any further. Let's say the converse is happening. You have high blood pressure in the high blood pressure. What's going on is there's little sensors in your atria, the top two chambers of your heart, that release this little guy. It's not anti-natriuretic peptide, it is atrial natriuretic peptide. Let me go ahead and fix that real quick. Nice little segue in, that's actually kind of nice because it really points out and gives the point that this atrial natriuretic peptide is coming from the atria. So what it does is it comes back on your capillaries and it makes them more permeable. If you make them more permeable, that means more liquid can get out. And if more liquid goes out, that means less volume in your blood. Less volume in your blood decreases your blood pressure. Now, we've got done talking about the glomerulus. Let's go ahead and focus in on that proximal tubule. Now, the proximal tubule is the guy that's like super duper greedy. If there's any proteins, amino acids, or glucose in there, it's going to grab it. If there's water in there, it's going to grab it. In fact, I've related it to being the most primitive or primal portion of your nephron. If it sees something delicious like meat, aka protein, or amino acids, or sugar, it's going to grab it and go, mine, 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 nom, 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 nom. And it's going to remove all that stuff. So it grabs, it has that primal urge to grab all those essential things. Now, something to remember about this is that primitive man has only so many hands to hold or so big of an arm to hold on to things. AKA, it can't, if you go over a certain limit with any of these things, you're going to see it in your urine. So for instance, I believe sugar will start showing up at 300 micrograms per deciliter. AKA, this is how they used to diagnose diabetes mellitus, is that the doctors would go ahead and smell or taste the urine to see if it's sweet. If it's sweet, guess what? Your insulin's not working, which means you have diabetes mellitus, which translated correctly, it just means sweet urine. So that proximal portion is primal and grabbing all those wonderful, delicious treats. Now the loop of Henle, this is the one that grabs and pulls out a lot of that water, a lot of essential, important ions for your extracellular fluids and matrix like your blood. It does so via a concentration gradient. The outer portion of that kidney, aka the portion of that medulla, we're actually in the medulla right now of the kidney. The outer portion of that medulla is somewhat concentrated with the sodium chloride, but down deep down in this like darkish brownish purple, whatever this color is heavily concentrated in ions, meaning that it really loves water down there, and it's going to pull out as much water as it can. So as you descend into it, this filtrate descends down this descending loop of Henle, or this arm of it. Water is going to come out. It's going to see all those salts and go, I like you. And it'll come out and get grabbed up and enjoy itself back into the medulla. Now, as it's going, that's what happens as it goes down. But as you go back up, something interesting happens. This little ascending portion of the loop of Henle is impermeable to water, mostly impermeable to water, which means as you go up, the osmolarity out here decreases, which means salt wants to come out. Sodium chloride will come out here. So here's a conundrum though. You got this going on, why doesn't this concentration gradient disappear? You got water coming out at one side, sodium chloride coming out the other. Why doesn't it just become equal on all sides? Why doesn't this just become a perfect concentration all the way through? Well, there is something going on here. There is actually a counterbalance. It doesn't seem like it makes sense to have this concentration gradient the way it is, but there is a counterbalance that is your blood. Your blood is the one that, once that water comes out, it's going to get absorbed into the blood. And when it's going back up, that blood is going to grab that sodium chloride. So it's grabbing the excess water and is grabbing that excess sodium chloride, leaving intact that concentration gradient. So good nutrients to medulla, keep medulla gradient. You're going to be A-okay. Now let's move on past the loop of Henle. We got through the descending loop. We just came up the ascending loop. We are now to the distal tubule. And this is the place where you see a lot of pH regulation. You see protons getting pumped in if it's too basic in the blood, or sodium chloride or what have you getting exchanged depending upon the status of the blood. It also has a lot of responsibility with potassium and calcium regulation. Now, how I remember this is I remember that right there. It's a distillery. It is very fine and refined, trying to make certain that every last bit of taste, that warm, yellow, foamy goodness, has the appropriate pH and balance. Delicious. So a couple of hormones that deal with the distal tubule. I call it the distal convoluted tubule just to make certain you remember that. The parathyroid hormone comes along, hits up that distal convoluted tubule, which If you're remembering, parathyroid hormone pulls out calcium from bones. Well, it's not just there. It's going to go ahead and pull calcium from this to try and resupply that rebalance that calcium. It's also going to kick out phosphate into the filtrate. We don't want that phosphate messing with the concentration gradient we got. Another hormone that messes with the distal convoluted tubule is actually branching off this guy. Remember that whole low blood pH type of thing, or low blood PP, blood pressure? Well, angiotensin 2 does one extra thing. It activates aldosterone. And aldosterone, what it does is it comes back, hits up that distal convoluted tubule, where it acts to pull out sodium from that filtrate. And if sodium is pulled out, guess what follows behind it? Water. Things that they want to get rid of as well, give that proton balance. This aldosterone has that role there, and it gets rid of that potassium. We don't want potassium in the extracellular fluid. We want it inside the cell. If it's outside, we need to get rid of it, or else we're going to have some dysfunctions. Finally, after the distal convoluted tubule, we have ourselves a collecting duct, which is extremely impermeable to water, unless and only in this condition is a hormone called ADH is present. ADH is called, in its fullest, antidiuretic hormone. Let's pull apart that word. Diuretics. Those of you that are nurses all know very well what a diuretic is. It's something that makes your patient pee more. Lots more. So if it's peeing a lot more, an antidiuretic would be the reverse of that. It is against peeing a lot, meaning that you are going to pee less with an antidiuretic hormone. This hormone is like the #1 hormone in water regulation and keeping your water inside your blood. Now, allowing this to happen, it opens up a hole called an aquaporin in these tubes. And water will chase after sodium in this sodium concentration gradient. Remember that this medulla, as you're going down, it becomes more and more concentrated with that salt, which means water is going to chase after it. So an acronym to help you remember is called always digging holes. Remember, it's making holes in that collecting duct so water can get out. Now, antidiuretic hormone, like I mentioned before, really does have a role in how concentrated your urine is. And important nomenclature I need to hit up on is if it's if the concentration gradient or concentration is synonymous, or you can exchange out the word specific gravity. If you have a high specific gravity, you have high concentration of urine. If you have a dilute urine, you have a low specific gravity. Specific gravity just asks, how thick is your salt in your urine? How concentrated is it? So with that, let's do a quick concept check. So this is your glomerulus/Bowman's capsule combined. This is your proximal convoluted tubule. It's very primal, grabbing up all those sugars, proteins, and amino acids, as well as a lot of your water. The loop of Henle with a descending branch and ascending branch is responsible for pulling out water as it goes down the descending loop, but also pulls out salt and as it goes up the ascending loop. And that concentration gradient is maintained by a counterflow of blood. This distal convoluted tubule is the distillery, where you make that nice, warm, frothy, delicious liquid and make certain that its pH is just right. Why did it go to a weird accent? I don't even know if that's even an appropriate world accent. Oh well. Finally, that distal convoluted tubule leads into the collecting duct, where it's extremely impermeable unless you have ADH, antidiuretic hormone. With that, let's apply a couple of our thoughts. If we were to go back, there's a couple of things you could run with. You could say that there are hormones, a lot of ADH going on, pulling out that water, but the main thing that's going on is right here. that loop of Henle. It is extremely deep in comparison to that of the human. Much deeper, so it pulls out a lot more water, which means its urine is very, very concentrated. I wonder if it has kidney stones. A question for another time. But with that, we are done with the nephron. Let's go ahead and look at the macrostructures of the kidney and the urinary system. It's about to get a little bright. I'm sorry. So here is your wonderful urinary system. You're going to notice you have your kidneys here. That's where your nephron resides. Just above it is the adrenals, the gland that adds to the renals. Kind of nice that they named it that way. Coming off of that kidney is the ureter right there, coming on down to the bladder. Bladder sits there, and then just below that is your urethra. That is the basic layout of your urinary system. If we go ahead and take a look-see at the other bits and pieces of your kidney that are important, first, you'll notice that it does look like a kidney bean. I wonder why they named it a kidney bean. Kind of looks like a kidney. Well, right where that seed would germinate for that kidney, you have the renal hilum. That is where the majority of the things that need to get into it and do its job innervates. So you got your artery, you got your ureter coming off, you got your vein coming off as well as quite a few of the lymphatics and nerves. Now, if we were to cut open that kidney bean and take a look deep inside, we would notice that first we have a cortex that lights light pink, which is anchored down into the insides by these renal columns. You'll notice that those blood vessels are spanning into the cortex and into these columns, but not into the medulla itself, or into these pyramids, I should say. The medulla has these little dark pink upside down pyramids. This is where that concentration gradient is that I was talking about. And sure enough, there's a little drawing of a nephron that's probably way too big for what it is. You got your glomerulus, you got your descending loop, and you got your collecting duct. Notice that they're both inside this renal pyramid. Now, I did mention that those collecting ducts come on down, and a lot of other nephrons will connect into this collecting duct. And as they do that, it gets larger and becomes a papillary duct. These papillary ducts will then form into this little triangular, open, very large piece right here that is continuous with the ureter, and that is called the minor calyx. Multiple minor calyx come together to make a major calyx, which is this guy right there, and a couple of major calyx will come together and form into the renal pelvis. You'll notice that that renal pelvis is coming pretty darn close to that hilum. Renal pelvis will then feed any of its concentration or all of its contents into the ureter, which will then continue on down into this glorious beast that is your bladder. It follows down into these ureters and comes into these ureterial openings. Now, couple of quick things. First off, this muscle in here is smooth muscle. You do not have control over it voluntarily. It is also lined with a transitional epithelium, one of its only in its kind in your body, meaning that it can get stretched, it can get pulled, and it won't have any detrimental effects on that epithelium to a degree with comparison to other bits and pieces of your body. Now, you do have a bunch of nerves innervating this bladder that senses that stretch. Usually, those nerves will give a reflex to your spinal cord, and the spinal cord will come on back and say, hey, you got to pee. And when you're a kid and you're not potty trained, it's an automatic thing. That internal urethral sphincter, this guy right here, just opens wide up, and that external urethral sphincter, that's skeletal, will just open on up and it's gone. You're just going to pee yourself. But as you train yourself to overcome that reflex, you're actually able to hold that urine. But that urgency is still there. When you hit about 200 milliliters inside your bladder, you get that sensation that you just gotta go, and you got to go now. And when that happens, the internal sphincter already opens up. Your external sphincter, when you're finally ready and positioned yourself into a place where you can relieve yourself, opens up and you get your liquids being secreted out your urethra. Now, the length of your urethra depends upon your sex. If you're female, it's nice and short. It's positioned next to quite a few different orifices along your body, while the male, it has usually about a 5 times in length difference to that of the female. It also has a couple of other extra tubes attaching to it, along with this little structure right here called a prostate, which we will get into when we talk reproduction. Now, the poor unfortunate souls that are females, you are more likely to get a UTI based on the positioning of these openings and how close your bladder is to your urethral opening is to the rest of them. The rate or how bad the UTI depends on how quickly that bacteria grows, as well as if it really likes that urine. Because that urine, if it's salty enough, can usually block from most bacteria from growing, unless they are halophilic, meaning that they love nice salty conditions. And if they're aggressive in growing, they can sneak up into your bladder and even on into your ureters and kidneys, which ends not very well at all. With that, that is the end of the presentation. Thank you very much and good luck.